But now to kick off this afternoon, we are really delighted to have our keynote presentation from Professor Chaitan Khosla. Professor Khosla is the director of ChEM-H and the Wells H. Rausser and Harold M. Head of PRiME Professor in the School of Engineering and Chemistry at Stanford University. His research is focused on two key areas: the chemical biology of polyketide synthases and the biochemistry of celiac disease, and particularly the role of tissue transglutaminases. His trailblazing work in these areas has been recognized with many awards, including the Waterman Award from the NSF, the Pure Chemistry Award from the ACS, and he is an elected member of the US National Academy of Sciences and the National Academy of Engineering. We asked Professor Khosla to be our keynote speaker this year because his groundbreaking research involves a multidisciplinary approach and forges important linkages between basic mechanisms of disease biology and the search for new therapeutics. Professor Khosla, we are so pleased that you are here today with us, and we are very much looking forward to your lecture.
Thank you, Sheena. That is so kind of you to give me a chance to come visit. And the only regret I have is that I don't get to come and visit your beautiful town and your beautiful campus and meet your wonderful colleagues and co-workers and students in person, but hopefully we'll have a chance in not too distant future. If I can share my screen, let's see. Can you see my screen?
Yes, we can.
Great. I first want to congratulate you on the launch of PRiME. It was an inspiring 20 minutes of an introduction, both from your university president and from Sheena Kelly on the progress that you have made, both in terms of bringing community together and building programs that impact not just academic research, but the character of the biomedical ecosystem in your city and probably even in your country. And so what I get to do in the next half hour is share with you some very analogous experiences that we're going through at Stanford in the context, through the lens of my own research on celiac disease over the past 20 years. And to do so, I want to introduce our version of PRiME at Stanford. It is a program we call the Innovative Medicines Accelerator that I was asked to help the university put together. It's a presidential initiative that was launched last year, shortly before COVID. And it has a very simple vision statement, which is to accelerate the translation of the basic science discoveries that happen on a campus like Stanford day in, day out into new medicines, while also expanding our knowledge of human biology. And what I hope to do is to give you a sense of how this works, but to give you a clearer idea of how we are trying to operationalize our President Mark Tessier-Levine's vision that I just stated in the previous slide. At Stanford, I want to introduce to you what I think all of you understand very well, that what we're all trying to do in our own ways on our campuses is bridge what people often refer to as the valley of death. And that valley is the one that connects the discoveries that are made in basic research laboratories across campuses like the University of Toronto and Stanford University to proof of concept studies in human patients that demonstrate the promise of these discoveries. And the Innovative Medicines Accelerator at Stanford imagines this valley of death as not one, but two valleys of death. There's the valley that connects the basic discoveries that our investigators make to a proof of concept in some disease model, typically an animal model, but in this day and age with IPS lines and whatnot, you can also have other types of valid disease models. But whatever is the benchmark disease model, when you have a hypothesis that was generated or a platform technology that emerged from basic research, the first testing ground is one of these legitimate disease models. And then there is another valley of death that lies between the animal model and the patient themselves. And so to bridge the first valley of death, we're building at Stanford what we call a drug prototyping pillar. This is a pillar that contains the capabilities and infrastructure that is needed to be able to convert core hypotheses or platforms into legitimate drug prototypes that can validate the hypothesis in a disease model. You will note I am using the word prototype and not drug. In some cases, this pillar of excellence may allow us, such as in the areas of cell therapy or perhaps in the areas of even protein therapeutics, to build legitimate drug candidates. But for the most part, what we envision these domains of excellence within the drug prototyping pillar doing is building the kinds of tools that will allow us, building the prototypes that allow us to validate key hypotheses. Because once you get there, industry and the investment community can understand how to take projects from that point forward. A key point I want to emphasize about what we're doing in the drug prototyping pillar is to build it in a disease agnostic manner. Now, the second valley of death is just as important. And that is the valley that we're looking to bridge with a pillar that we call the experimental human biology pillar. Like the drug prototyping pillar, this contains domains of capability and infrastructure that allow us to do the studies in human subjects in a safe, resource efficient, and ethical manner to be able to test hypotheses that come from the bench, but equally importantly, allow observations on the bedside to motivate future science and engineering on the bench. And so the experimental human biology pillar at Stanford includes capabilities that allow investigators who are not traditionally in the habit of considering the human patient as a research subject to be able to design studies and implement them. And these can include capabilities like a CTRU to be able to support studies. This is something that has served us very well in the COVID pandemic, where we built a COVID-19 outpatient CTRU that has done multiple clinical trials through investigator-initiated INDs. As well as other capabilities that we have to be able to support this kind of research. And obviously, these two pillars do not stand in a vacuum. They are built on a foundation of excellence that comes from the institute I've had the privilege of helping the university build, called Stanford ChEM-H, and of course, our even greater and broader strengths that exist within Stanford Medicine. So this, in a nutshell, is what Stanford's Innovative Medicines Accelerator is. And in the remaining time I have, what I want to do is to share with you how my own experiences in the context of celiac disease over the past couple of decades have given me a lens to think about the opportunity that the IMA presents to a university like Stanford. So celiac disease is an autoimmune disease that occurs in about 1 in 100 patients, 1 in 100 humans in most parts of the world. And while there's many things I'd like you to know about this disease, if you've never heard of it, for the purposes of this talk, there's 4 things you need to understand. The first one is that the primary target of this autoimmune disease is the small bowel, although other organs, as you see in the right slide, where this patient has a skin manifestation of celiac disease called dermatitis herpetiformis, other organs can be affected as well. Like most other autoimmune diseases, whether you think about type 1 diabetes or rheumatoid arthritis or multiple sclerosis, this disease also has a T cell and a B cell component, both of which are quite well understood and characterized. As I mentioned, it's a widespread disease. It's a lifelong disease. And what motivates us to pursue research at the chemistry biology interface is that even though the disease was first characterized in the medical literature more than 60, 70 years ago, even today, there is not a single alternative, therapeutic alternative to the only form of treatment that exists, which is a strict lifelong exclusion of all forms of dietary gluten. To give you a sense of what 20 parts per million in a diet is, it's about the amount of gluten that is present in half a crouton. So this presents an extremely challenging condition for patients with this lifelong diagnosis to manage, and creating alternatives, therapeutic molecular alternatives, is an important goal for us and for others. So at the time when we started research on celiac disease, this slide summarizes what we knew about the pathogenesis of celiac disease. It was well known that dietary gluten was the primary environmental trigger of this disease. And on the other side of this disease, it was also understood that there were disease-specific T cells that existed in the small bowels of celiac disease patients that had all the hallmarks of autoimmune disease driving T cells. These were gamma interferon secreting inflammatory Th1 cells that were eliciting activity, that were being activated by gluten-derived epitopes. And between these T cells and the primary environmental trigger lay a couple of key processes and molecules. So as you eat gluten, it goes through your GI tract. It breaks down first into peptides and ultimately into amino acids. And some of the peptides that are generated through this digestive process are modified in a regioselective manner by an enzyme that exists in the gut and in other organs called transglutaminase 2, or TG2 for short. What this enzyme does is it converts specific glutamine residues in dietary gluten-derived peptides into glutamic acids, also an amino acid, nutritionally important amino acid. But it's the modification of these gluten peptides that allows them to be presented on HLA-DQ2, which is the primary disease-associated major histocompatibility complex protein in celiac disease. So more than 95% of celiac patients express HLA-DQ2, and it is HLA-DQ2 that was known to be presenting these modified gluten peptides to the disease-specific T cells. There were also autoantibodies that were generated in the disease, and it had been known around the time we started working in this that these autoantibodies are generated toward the human protein, TG2 itself. And so our research over the past couple of decades has been motivated by three simple questions that you can ask if you look at this pathogenic model through a molecular lens, which is, of all of the dietary proteins we consume on a daily basis, what's special about gluten? The human HLA repertoire contains literally dozens, if not hundreds, of different HLAs. What is specific about HLA-DQ2 that allows it to be such an important player in celiac disease? That was another fundamental question. And then the last question had to do with why TG2, this transglutaminase enzyme that post-translationally modifies these gluten derived peptides. The answer to the question why gluten came relatively early in our research program. It turns out that dietary gluten is unique among all dietary proteins that humans consume, in the sense that a vast majority, there's distinct portions of, so what I'm showing you in this slide are two sequences, one-letter amino acid sequences of two distinct gluten proteins that you would find in a bag of flour if you went out and bought a bag of flour in a grocery store. And what you see is that these are relatively proline and glutamine rich sequences. We can discuss why that is the case later if that's of interest to you, but for the purposes of the question why gluten, what you need to understand is that these kinds of dietary proteins derived from gluten are unique because they have large chunks of them that are virtually untouchable by any of the GI proteases that you've learned about in textbook biochemistry. So when you think about enzymes like pepsin, trypsin, chymotrypsin, as well as all the brush border proteases and peptidases that exist in the small bowel that are responsible for taking dietary proteins and breaking them down into nutritional amino acids, those proteins do not modify the red portions of these sequences. The net result is these sequences persist for substantial durations in your small bowel. And this is true not just for celiac patients, this is true for all of us. So those of you who had a sandwich for lunch before this or something of that sort, if you had a ham sandwich, the protein in the ham and cheese by now has been completely broken down and absorbed as amino acids, whereas these portions shown in red that are in the bread of your sandwich will probably persist as intact peptides for the next couple of hours in your intestinal lumen until they finally encounter the bacteria in your lower intestinal tract. And it's no surprise that these portions that are proteolytically resistant are also the portions that are most immunogenic. So gluten is unique because it creates metastable peptides for the immune system to recognize. And that led us to a hypothesis that we could treat celiac disease by engineering an enzyme that would break down the the metastable peptides, thereby accelerating what is the kinetic bottleneck in gluten proteolysis in humans. Fast forward 15 years, there is now a protease that was engineered in our labs in the mid-2000s called latiglutenase that has gone through clinical trials at many centers across the world and continues to go through clinical trials that I might come back to later in my talk if time permits. The question, why HLA-DQ2 and why TG2, the answer to that came from structural biology. This is an example of a picture that is really worth 1,000 words. Because what you're seeing in this picture is the crystal structure of human HLA-DQ2 bound to the immunodominant or one of the immunodominant epitopes that is present in dietary gluten. And what you see is a couple of things. The first thing you see is there are many prolines in this sequence, which is somewhat unusual in the context of MHC epitopes, because most MHC binding of peptides happens through backbone side chain, backbone interactions. So hydrogen bonding on backbone amide linkages. And when you have prolines, that limits your ability to form hydrogen bonds on peptide backbones. And so what you're seeing over here is HLA-DQ2 has a register that allows prolines to exist without penalty in a handful of locations. And somehow the gluten in the wheat fields has evolved to take advantage of precisely that pattern. So that's the reason why HLA-DQ2 is special. There's a second reason why HLA-DQ2 is special within the human repertoire of MHCs, and that brings in TG2 as well. This glutamic acid that you're seeing over here that beautifully salt bridges to this unique lysine that is present in HLA-DQ2 did not exist in the wheat field. This glutamic acid was generated post-translationally in the intestine by TG2. And so that tells us that that post-translational modification has a profound relevance to the pathogenesis of celiac disease. So these kinds of insights led us to recognize that while clearly both HLA-DQ2 and TG2 are critical to the pathogenesis of celiac disease, one of these protein targets is probably going to be very difficult to drug, whereas the other one might be druggable. And so we focused starting then on the goal of making TG2 into an appropriate drug target for celiac disease. How do you go about that? Through chemical biology, you build tool molecules that allow you to do things. And so what I'm showing you in this slide is a tool molecule out of many that we built to be able to visualize when TG2 is active. So this red color over here, essentially when you inject a mouse in its tail vein with this red compound, what happens is wherever TG2 in the mouse is active, it lights up with the red compound. And what you're seeing is in all these panels, you're seeing a lot of TG2 in the mouse intestine. That's what's being stained by the antibody against TG2. But only in this panel are you seeing rapid turn on of activity. The second tool molecule that we built was a TG2 inhibitor that allowed us to confirm that this this putative activity we were seeing was indeed coming from TG2, because when we co-administer these two molecules together, this inhibitor blocks the activity of TG2. And so what tool molecules like this did for us was it taught us something that we weren't expecting at the level of TG2 biology. The human gut is chock full of TG2 protein. And for the longest time, it was assumed because there's a lot of TG2 protein, it's no surprise that when gluten antigens show up, they can get modified. However, what the data in this slide says is that in a healthy gut, mouse or human, there isn't much TG2. And so somehow this TG2 must be turned on in the intestine, which led us to a hypothesis that it is gluten-induced inflammation that induces TG2 activity, leading to a vicious cycle type of process where the T cells activate gluten, activate TG2, and TG2 generates more T cell activity. It only took us about 10 years to test that hypothesis. So this is an experiment that was done recently, not too long ago, but in collaboration with Bana Jabri's lab at the University of Chicago, because Bana had built a mouse model of celiac disease, which is beyond the scope of what I can describe. But what I'm showing you over here is that the mouse model shows the same characteristics, except this time it's showing it with dietary gluten. So in the absence of gluten, when the mouse is put on a strict gluten-free diet, there's not much TG2 activity in the gut. But when the mouse is challenged with dietary gluten, TG2 activity turns on. And by this point, we had good mouse-grade tool molecules, drug prototypes that block TG2. And so we could confirm that this activity we were seeing in red was indeed being generated from TG2 in these celiac mice. And that allowed us to do the critical experiment. So it took us literally 10 years of tool building and model building to be able to answer the very simple but profoundly relevant drug discovery question, which is, if you block TG2, can you achieve clinical benefit? That was the core hypothesis we were at in mid-2000s. It took us about 10 or 15 years to confirm that if you block TG2 in the presence of a gluten challenge, you can prevent villus atrophy, which is the clinical hallmark of celiac disease. And so this allowed us to further invest in optimizing compounds. By us here, I'm referring to startup companies that were built off the tool molecules licensed from Stanford. And today, those molecules have moved forward into clinical studies by GlaxoSmithKline. So where we are going right now is to answer the core question that we started with, back to the basic science drawing board, which is, how does this reverse connection between T cells and TG2 happen? We need to understand that, because presumably that will teach us how TG2 is activated in the earliest stages of disease, and we can study the onset of autoimmunity. In the last 5 minutes that I have, I want to also share with you progress that we've made over the years to expand, to use our growing insights in celiac disease in the context of the human experimental biology pillar, because that is just as important an area where molecular science can have an impact. And the relevance of experimental human biology in the context of celiac disease became apparent to us about 15 years ago as latiglutenase was approaching clinical trials, which is, how does one go about testing a drug candidate in a disease where there's never been a clinical trial for any drug before? are you going to measure in order to be able to tell whether a drug works or not in a patient? And the benchmark back then for diagnosis and disease management is something called a small bowel biopsy. There's a quantitative measurement that can be made on a small bowel biopsy that I don't have time to go into, but what it does is it allows you, this measurement changes if you take a celiac patient whose disease is in remission because they've been on a strict gluten-free diet for a long time, and you challenge them with modest amounts of gluten, 1.5 grams of gluten for a few weeks, 3 grams of gluten, 6 grams of gluten for a few weeks, just to give you a sense, about 1.5 grams gluten is what you find in a typical slice of bread. So what you're seeing is that this parameter shifts down. And that is the gold standard for saying that somebody is a celiac patient. This is an extremely powerful measurement. In fact, in the first phase 2 clinical trial that was done to show that ladiglutenase does actually work as a proof of concept in patients, it was the small bowel biopsy that proved that, because these patients who took placebo drug showed relapse on their biopsy, whereas the patients who took ladiglutenase didn't, and the p-values over here were very statistically significant, about 0.01. But it was not a viable way to do future longer-term clinical trials, because these biopsies are invasive, they're variable, there's a small dynamic range, lots of standard error, very difficult to implement in multi-center clinical trials. And above all, even if you could somehow limp across the drug approval finish line with this kind of a thing, using this kind of a metric in clinical practice on a day in, day out basis would be exceedingly difficult. So it was pretty clear we needed better surrogates. Where are you going to find these kinds of surrogates? Our first crack at finding a surrogate was to look at molecular insights that come from celiac intestines themselves. In particular, we learned through the literature that CYP3A4 is an enzyme that undergoes significant repression in an active celiac intestine, but then returns to normal activity in celiac patients. And this motivated us to develop a surrogate marker that allows us to use a widely used oral medicine called simvastatin. It's a safe medicine that undergoes CYP modification and leads to a big change in the bioavailability of the drug. So the change in this bioavailability became an endpoint in itself, a surrogate endpoint for disease activity in patients. So what you're seeing over here is a multi-center study that was done in New Delhi, Zurich, and in the US that looked at about 40 patients over 4 years to be able to show that there is a strong difference in the activity of in the levels of plasma simvastatin when you give patients a single dose between healthy volunteers where it's low. So that would be analogous to this curve over here. We are measuring this level of simvastatin, whereas in celiac patients, that number is going much higher because their intestinal CYP3A4 goes down. And as you treat patients over time, these levels come closer to healthy volunteers. And so this marker is now being tested side by side with the gold standard in a phase 2 study at the Mayo Clinic involving ladiglutenase. This illustrates the importance of having companions. More recently, we've been using these immunotoxic peptides, and the fact that now there are commercial tools to be able to test for these immunotoxic peptides in food by using like dipsticks and things like that, It became clear recently that you can take these same dipsticks that are supposed to check for whether your yogurt contains gluten or not. You can dip them into human urine, and if that person had gluten, that dipstick lights up. That got us interested, and so we decided to ask a fundamental question that, to my knowledge, has never been asked in humans, which is, do intact dietary peptides from our food actually make it into circulation in human body? To do this, Brad Polonsky and Nielsen Weng teamed up with my colleague Nielsen Fernandez Becker to develop a protocol that essentially showed not only can you identify immunotoxic peptides in the circulation of people who have taken gluten, but celiac patients tend to have many unique peptides that you don't see in healthy volunteers. And that opens up a really interesting vignette into what this disease might teach us in the future. I think in the interest of time, I should stop over here. What I want to re-emphasize one more time is that trying to bridge these valleys of death is not just an engaging and highly intellectually stimulating and hopefully also rewarding exercise, but it also gives our trainees an opportunity to learn from scholars from many different fields that no single PI can provide. And this is where I feel incredibly grateful that the students and postdoctoral fellows in my lab who have worked on celiac disease and have pursued projects have benefited from the wisdom of many of our clinical, translational, and technological collaborators over the years. Last but not least, I would be remiss if I did not congratulate the PRiME fellows who we are looking forward to hearing next. And I thank you once more for your very kind invitation to come and talk to you about celiac disease and Stanford's Innovative Medicines Accelerator. Thank you.
Thank you so much, Professor Khosla. I wish we could now give you a thunderous round of applause. It was a wonderful talk, very inspiring and amazing story. And I want to encourage people to put their questions in the Q&A box, and we'll bring as many of those forward as we can. I've collected up a few questions over the course of the lecture that I'll go ahead and ask now. There was a question about what's known about the genetic basis of celiac disease and how that may fold into some of the observations that you've made biochemically.
That's an excellent question. There's a couple of different levels at which I can answer that. The first level is the one that's probably most relevant. If you have celiac disease and you have an identical twin, your identical twin has a greater than 2 in 3 chance of getting celiac disease within their lifetime, which tells you that there is a very strong genetic dependence, but it's not strictly Mendelian. So there is an environmental component as well. Now, where is this genetic dependence coming from? About half of it, if you think about how geneticists analyze genetic dependence, they usually assign load scores to individual loci. About half of it can be assigned to HLA-DQ2. So we know that HLA-DQ2 is necessary to get this disease. The handful of people who have celiac disease but don't have HLA-DQ2 have HLA-DQ8, and by now there's very good evidence that the story I told you about HLA-DQ2 is entirely applicable to HLA-DQ8. But what's the rest of the other half? There's no single locus that explains that. And so that's where the future of celiac genetics is taking the field. What are all the other loci out there that are allowing us to see this growing level of celiac disease in the population that are contributing to it. My own theory, but at this point it's strictly a theory, is that those are the loci that somehow allow a higher baseline activity of TG2 in people, in pre-celiacs, and that somehow when they encounter gluten, that higher than average threshold leads to the level of inflammation getting to a critical point that when T cells evolve, now you can start having a vicious loop going. But that's pure theory right now.
Yeah. Okay. There's a related question that's just come through regarding the triggers of celiac disease. So there are theories out there that there could be a viral infection that triggers, or other things. People eat gluten most of their lives and then one day start metabolizing it differently. Do we understand what's going on there yet, or is that still really
Yeah.
unknown?
The short answer is the theories on which that the field was betting on for about close to 2 decades have been completely dashed as a result of that. Two very large studies that were done, one in Northern Europe and one in Southern Europe, that was subjects of 2 recent New England Journal papers. Basically, the simple answer to your question is we have no clue. The best hypotheses that we have turn out to be wrong, and so we're back to the drawing board.
Okay, well, then more interesting questions to be answered there for sure. Here is another question. Can the peptide be degraded by live medicine? So for example, a microbe that could be engineered to degrade it so that that then allows the peptide to be kind of disposed of.
Yeah, that's a brilliant idea. And if I live long enough, I would hope that some wise and highly ambitious translational medicine researcher in 2040 is on the cusp of testing that hypothesis in human patients. That will require greater control of the microbiome if you want to test that hypothesis rigorously than we currently have. The key point I want you to remember as you think about how to attack that challenge, which is a very interesting one, is this is a disease of the upper intestine. There are a lot of people who interrogate the celiac microbiome, but the vast majority of those studies involve interrogation of the stool. The challenge you have in those studies is how do you interpret what is happening by the microbiome in the duodenum when a vast majority of what's in the stool came from the ileum and the colon. So
Yeah.
that's a challenge.
Absolutely. Okay, maybe one more question here. There's been lots that have come up. Does TG2 provide an essential activity in the gut other than the modification of this peptide? And there are also some questions about, is there cysteine as a nucleophile, and could there be suicide inhibitors that get developed?
Yeah. So the answer to the latter question is yes, cysteine is the nuke. You should think of TG2 as essentially a cysteine protease in drag. It's hydrolyzing a side chain amide instead of a backbone amide. And you can play the same games, and we and a lot of other people do. Your first question, does it involve and provide an essential activity? That is obviously the nub of the issue. What got me interested in TG2 as a drug target in the first place was the fact that 2 independent labs had engineered TG2 knockout mice. These are not just gut localized knockout, these are whole body localized knockouts. And both those animals to first order were non-phenotypic. They live, breed, develop normally. And so that gave us confidence, and our pharmacological tool molecules over the years have more or less confirmed that, is that this is a non-essential activity in the body, not just in the gut. And so if you block TG2 in an intestinally restricted manner for the duration that food is traversing the gut, hopefully you can get therapeutic, a decent therapeutic window for a lifelong disease.
Yeah.
Good question.
Okay. Wonderful. Well, thank you so much, Chaitan. This was just a wonderful lecture to kick off our symposium with. Absolutely inspiring. So wonderful to see you again. I wish there was not a bunch of electrons between us, but that you were here with us on campus. But we will do that another time. And again, thank you so much for inspiring us all. So with this, we're going to now turn